Now, let me introduce the speaker of the day. Welcome Dr. Binoy Rajan, who works as clinical scientist in Norway. He did his BFSc from College of Fisheries, Panangat, MFSc from CIFE, Mumbai, and PhD from Nord University, Norway, on mucosal proteomics of Atlantic coast. After his PhD, he worked as a few months as faculty at Kufos, Kerala, and then moved on to a postdoc position at the Norwegian School of Veterinary Science, exploring passive immunization of Atlantic salmon. The last 4 years, he has been working as clinical scientist in Pharma in Oslo, Norway, which is a subsidiary of Zoetis USA. He works on developing vaccines and therapeutics for fish. FYI, Pharmac is the largest vaccine maker in aquaculture, mostly for salmon. Today, Binoy will be talking to us about developing and implementing vaccination for farmed fish. Binoy was my junior in College of Fisheries, Panangal, as well as in CIFE, Mumbai, and I'm meeting him after many years, and I'm indeed glad to host him here in AFS Talks today. So welcome again, Binoy, and the stage is yours.
Yeah, thank you, Sajana. Thank you for the kind words. I hope you can hear me loud.
Yes, you are clear.
Yeah. Yes, and thank you also for AFS Talk for having me. I can try to share my screen now. Yes, and I hope you are seeing the screen.
Yes, your screen is visible.
Yep. Great. Yeah, so as Sajana mentioned, I'll be giving a talk about fish vaccination, how a vaccine is developed and how it is implemented. This is supposed to be a technical guidance, but please remember that there will be a lot of theoretical consideration as well. So I don't expect any of you to start a vaccine business tomorrow, but so it will be more like a bit of an overview and some insights into the practice. Just to put into context, why the big question is why we need to vaccinate fish. We are living in a time where we are slowly understanding the importance of vaccination, or at least getting a feel of it. Of course, the main aim for vaccination is to prevent the disease, but let me put it in another context. I'm working in Norway, and here fish farming and aquaculture is a very big business. It's a bit of a high-tech business. So, and also the mainstay of the farming is Atlantic salmon farming. And if you see this graph, let me just, yeah. If you see this graph, on the left axis is the amount of antibiotics that goes into the aquaculture in Norway. And on x-axis, you have the years starting from 81 to 2014. On the right y-axis, you have the biomass of the fish produced over the years. So you can see that the fish production has grown considerably. And that's not surprising because we know that aquaculture is growing at quite a faster rate compared to other agriculture aspects. If you look closely, I have just tried to put some important key events, actually. Initially, in the early 80s, when the farming took off, there were a lot of diseases. You had a lot of bacterial diseases, primarily, which were very devastating. And towards the late 80s, vaccination was introduced. And for instance, here, 87, Vibriosis vaccine was introduced. In the 90s, furunclosis vaccine was introduced. These were big, challenging diseases at that time. And slowly moving forward, there were a lot of vaccines available and a lot of multivalent vaccines. That is, a lot of antigens combined together in a single shot. And that has made it much easier So what you can see at the overall picture is that the antibiotic usage has gone down dramatically, and that is a big plus for the industry. Of course, 100% of this is not because of vaccines. There are also some other factors like improvement in technology, biosecurity aspects, more knowledge, etc. But vaccine has been having a primary role in reducing antibiotic usage, and that is a big plus for the industry because that raises the quality of the fish produced and always commands a better price. So if you are in competition, you command a better price if you, for example, if you don't use antibiotics, isn't it? So just to put that into perspective here, what is vaccination? Don't need to define it really because we are very familiar with it now. We are undergoing through that process ourselves. But just to define it, vaccination is a very simple and safe and effective way of protecting you against any harmful disease before you come into contact with them. It uses your body's natural defenses to build resistance to specific infections and make your immune system stronger. How they work is that vaccine train your immune system to create antibodies. Just like a COVID vaccine, it trains you to be prepared for a disease. However, because the vaccines contain only kill or weakened, or in the case of COVID vaccines, a nucleic acid like a mRNA, or in some cases live vaccines as well. So the risk of getting an infection is very, very low, almost nil. Going back to some fundamentals, defining immune response and what is immunity, etc. Immunity is the body response to an infection, and it can be broadly divided into innate and adaptive two arms of the immune response. The innate immunity is something that happens very quickly. It's the body is always ready for the invasion. But that has some disadvantages in that it is not very specific. It's like a broad measure by the body, like raising the temperature of the body, etc. So it doesn't serve a specific purpose, but it tries to remove any pathogen by the same method, all the pathogens by the same method. And also remember that there is no memory to this kind of response. And once you are through this innate aspect, you come to the adaptive aspect, where the body is trying to learn to differentiate between the different pathogens that it's encountering. And that provokes a response, and that response is sort of kept in the body as a memory cell. It remembers next time when the same type of infection comes. In the case of fish, as fish is our subject today, fish also is a lower vertebrate. It's kind of at the bottom of the vertebrate section, but it does have adaptive and innate immunity. So we have a possibility to vaccinate fish. Innate immunity in fish is, of course, physical parameters like the body, the skin, the mucus, the scales. The general body surface is a barrier for pathogens, so it's not easy to get into the fish. There are cellular components of the innate immunity like phagocytic cells and some other type of cells. Also, you have some humoral components like cytokines or antimicrobial compounds. These are all non-specific compounds. Then you come to the adaptive response. It's quite comparable to humans, even though it's not that broad, or there are no, it's not so advanced. So it seems fish do produce antibodies in response to an infection or a vaccine. That is a good thing. So we have something to work with. If you look at the timeline of immune response, just as I mentioned, when the fish or human is exposed to a disease, you have a microorganism that you are encountering, but it is initially in an incubation phase, and it starts to increase in number, and it reaches a level where the body suddenly recognizes, okay, now I have a problem. And that recognition is sort of a threshold level. And above that, the body starts to think, okay, now I have to respond to this infection, and the body starts to mount an infection. And by that time, the microorganism, the pathogen, would have grown exponentially. It will just grow, and by the time body is ready to fight back, it would have reached almost the top, and then the body is fighting back, and the pathogen is eventually removed from the body. So the initial phase is, of course, this innate aspect where it's very quick response from the body. Most of the time, it is not enough. In some cases it's enough. It could also depend on the amount of exposure you are getting. And a typical example is, of course, those who are fighting COVID in the hospitals, doctors, they tend to get a higher exposure than normal human beings out on the streets. So naturally, their chance of getting a disease is much higher because of the dose. So this cycle is quite interesting because let me just take this off. So the goal of vaccination is essentially to simulate this kind of infection and create this adaptive part with the memory cell so that the body remembers this, which will be useful when the actual disease comes. So fish are quite special if you compare them with humans. The key difference is that the body temperature of humans are stable. In the fish, being cold-blooded, it's very, very dependent on the water temperature, so it can fluctuate quite a lot. And therefore, the physiological aspects of fish are in a sort of way regulated by the environment. Immune system is also less advanced compared to humans. And when you think of vaccinating a fish or a human, in human, you know that it's usually we do an intramuscular injection. In fish, there are many possibilities. You can immerse them in vaccine. You can inject them into their body cavity, into the belly, or even intramuscularly inject them. Also, what is the response to the vaccination also is quite different because humans don't fluctuate in their body temperature. You can expect a stable response time. Like, okay, I give this vaccine, I get a response in 2 weeks. That's a given, roughly. In case of fish, this again depends on the water temperature. In the warm water species like tilapia, you probably get very quick response, 2-3 weeks, you get a good response. But in case of salmon, for instance, it will take a lot of time because it's in cold waters like 6-8 degrees. There's also a quite big difference in terms of adjuvants. In fish, it's very common to have a water in oil emulsion as an adjuvant. I will come to that later, but that's very, very much a backbone of modern fish vaccination. So what is an ideal fish vaccine? Let's just try to summarize, or at least come to an idea of what is needed. Because that is very important. We have some kind of an idea where we want to be if we want to develop a vaccine. And this is probably applicable to not just to fish, also many other organisms. But we take it for the fish for the moment. The vaccine should have induce good immune response. It should have a broad spectrum effect so that it ideally, because if, for example, you're developing a vaccine against vibriosis, you would want that vaccine to perform against many different strains and serotypes of vibrio causing bacteria, vibriosis causing bacteria. You will also, if preferably, have a cross protection against some other types. But at least within various types of vibrios, it should give some protection. And then you can maximize the benefit of the vaccine. It should have a long duration of effect. And that means you take different fish species, you have different production cycles, right? Tilapia, for instance, you farm them maybe like 4 months, 6 months, or even 9 months. There are some countries which go even further because they would like to have bigger fish. And and in case of salmon, it is at least 1 year in sea in cages. So there are a lot of variation between fish species, which means that the vaccine that you're developing for a particular type of species, it should cover preferably the whole life cycle or the production cycle, and of course we should preferably use only one dose or minimum number of doses. And those of you who are working with farm will know that how inconvenient and stressful it is for the fish to be taken out, injected or immersed, crowded together, etc. So we want to avoid that as much as possible. So a one-time injection or a one-time administration is the best solution, but that is the point where we want to be. And we don't want to stress the fish a lot of times during the production cycle, because that can cause other problems. And above all, the vaccine should be affordable, needless to say that, and safe for the fish and for human consumption as well. Yeah, so I have tried to sort of list out a lot of steps in this vaccination development, or developing a vaccine idea. It's not very exhaustive, and it is not as simple as it looks. There are a lot of hurdles, and it's a process. And if you want to develop a very good vaccine, and if you want to be sure that it is very effective, it's quite safe, etc., it's going to take a lot of time, maybe 5-6 years of research and development. So there are some other ways of doing things, but of course this will also depend on the country you are working in. So before you start a vaccine, let's go through some initial consideration. The disease itself, if you are targeting a disease, you know that there is a disease problem in a particular area, or maybe in a wider area, you have to evaluate how severe is the disease, what type of disease it is. Is it a bacterial disease or a viral disease? And how does this disease manifest? Does it cause a lot of mortalities? Are fish, like, do you expect that the fish, all of the fish in the farm dies, and that's almost 100% or 50%? Does the disease, on the other hand, maybe not necessarily result in a lot of mortalities, but it persists in the population and sort of affects the growth, and that it's like the fish are not growing at all. So it's essentially the same problem for the fish farmer. So you have to gather a lot of information regarding this, and because those are the parameters that you want to try to implement in your development process. Availability of healthy fish seed, this cannot be stressed much more than this, because it's one of the most important aspects in any kind of fish clinical study. Because you want to develop a vaccine, for instance, then you have to go to your wet lab or a lab where you can do controlled studies. That is the point here. But then you need to have fish for this, and that fish, it's very important that this fish is healthy. It's not already burdened with a lot of disease when you are trying to vaccinate it and test it. That would probably confound your results, and you will face a lot of problems. So the most important aspect is, of course, to have healthy fish seed. And this also applies to the end user. Finally, you have made a good vaccine, but the farmer that is using it is actually injecting it in a very unhealthy, or it has a lot of parasite, for instance, or so the fish is not good when it's being injected with the vaccine. So then you essentially are losing the effect of the vaccine. So it's very important that healthy fish seed is available, and followed by good screening and biosecurity practices. One of the key overriding things to all of this is, of course, the regulatory authorities, or I would say the government itself, has possibly delegated this to some agency within the government to monitor this development, to give license, etc. And they are essentially the people who are setting guidelines, what kind of tests you should do for the vaccine, how it should be developed, etc. What should be the parameters for the vaccine, etc. So that will decide in a way how you develop your vaccine and how long it takes for you to develop, because usually there are a lot of hurdles and it's justified because the government doesn't want you to make a vaccine that's very unsafe for a fish, for instance, or unsafe for the user, or even make a product that is not what it claims to be, right? So it applies to all kind of products. Yeah, so we have those aspects. I'll go to the next consideration, that is the type of vaccine. Well, those of who we are working with fish already might have read this in several occasions, and that is there are different type of vaccines. It has been a hot topic these days because of the COVID vaccine. So what I can tell is that if you think about type of vaccines for fish, you start with the sick fish, the problem fish. You try to isolate whether it is a bacteria or a virus, and then you try to propagate it in your lab, right? You try to grow it. If it's a bacteria, possibly using a media, or if it's a virus, you use cell culture to propagate it. If you are making an inactivated vaccine, that is where you grow the virus or bacteria, you inactivate or kill it essentially with formaldehyde, and then you formulate it into a vaccine. This is, for instance, like your Covaxin that we use in COVID vaccination. That is essentially a killed inactivated vaccine. And that is a very good way of doing this. It's very fast process because you just grow the bacteria virus, you kill it, and then you formulate it and with an adjuvant, etc., and then you inject the fish. So it's a very straightforward process. This is also very attractive for fish because the costs generally are less in developing such a vaccine. But in some instances, you cannot have any effect or any use of that kind of vaccine. Sometimes it might not work. And then you have an option of having maybe a live attenuated vaccine. That is the similar process where you try to grow your pathogen or the virus or the bacteria, but instead of inactivating it, you try to manipulate it. And that you can do in many ways. You can do several passages, like keep on growing it, and then one day you would expect, okay, that was very less virulence because the pathogen is adapted to cell culture or the media, and it's no longer, it's losing its virulence factors, and that way this virus or bacteria can be injected live into fish and still not cause any disease, but it can stimulate the immune system. So it's a good strategy, but it's a kind of a blind end because you're hoping for the best. You're passaging a lot of times and just hoping for the best that it turns out to be a non-virulent organism. So that's a lot of it can take a lot of time. It might take less time. Another way of doing is you manipulate the genome of this organism. You go in, you remove a virulence factor from the genome, and that way you can get an attenuated vaccine. The problem here is that if as soon as you start to tinker with your organism, you try to put some gene or take out the gene, it can get labeled as a genetically modified organism. And that means you have further hurdles to convince the government and other people, other stakeholders, environmentalists, etc., if this is a good idea to do, if you want to take this vaccine forward. So there are hurdles there as well. Another option would be to use a genetic substance directly, make a DNA vaccine, clone it into a plasmid, inject it into the fish, and that can be a DNA vaccine. I won't go into much detail here because this is all available and we already are very familiar with certain aspects. In fish, at least, inactivated vaccine is the big player. There are some one DNA vaccine, there are some live attenuated vaccine. Other type of vaccine also exist, other possibilities, but it's not quite relevant for fish as of today. Yeah, let's focus on to inactivated vaccine then, because that is at least 80% of the vaccine in fish industry today is inactivated vaccine. It's widely used. And what is this inactivation? Inactivation can be done in different ways. You grow a pathogen, an organism, a bacteria, virus. You can, for instance, inactivate using treatment with formaldehyde, and then you remove the formaldehyde by filtration, etc. And this antigen, or what you call the dead whole bacteria or virus, that is then added into an emulsion. An emulsion, it's like two liquids, immiscible liquids, are when they are mixed, they become an emulsion. If you know, if you remember your school studies. So what is this essentially forms is that the antigen is actually in water phase, in water, and then it's mixed and blended with oil. So what happens is that the bubble, the water droplets are captured in the oil media, and inside the water droplets you can have this our antigen, which is the whole killed bacteria or virus. In this case, for instance, let's say bacterial fragments are also there. Whole bacteria is also there sometimes. So this is like this kind of emulsion is the vaccine. It's a formulation. So how this is quite useful, and there is a theory. I mentioned earlier that in fish, the immune system is probably less advanced compared to humans. And a lot of studies have shown that fish doesn't have, or at least is not very having a robust memory cell aspect. So you inject a vaccine, maybe after 2 months, you don't find any effect. So, but you know that in fish farming, we want that duration of effect. So this emulsion, this oil emulsion, this helps in doing just that, because the oil, there is an oil coating, and there is water droplets, and there is antigen inside it. And this functions actually with the injection vaccine, I should say. When the fish are injected in the belly, this oil and this emulsion is deposited in the belly, in the peritoneal cavity, and it stays there because this is not degraded so fast. If you are putting your antigen just in water and injecting into the fish, maybe it will be degraded. Antigens will be taken up into the kidney or spleen, and then it will be destroyed. It will be discarded, broken down. But in this case, the oil layer helps to protect this antigen, and it stays there for quite a long time. And the antigen is slowly broken, like it's slowly released, and the immune system is recognizing it every time. So even after 10 months or 12 months post vaccination, you can still see this antigens in this with immunohistochemistry in the fish tissues, and that means it's still stimulating the immune system of the fish. And so there is no wonder here that when this vaccine is administered, you have a very high antibody response for a long time. So this, in a way, solved the problem for fish vaccine, because then you can really have a long duration of protection. And that is a big achievement for the fishing industry, because the longer the duration, the better the investment in the vaccine. When it comes to the live attenuated DNA vaccine, I also mentioned that some of the fish vaccines are using it. There can be instances where your pathogen is a intracellular bacteria, for instance, or a virus that grows within the cells. And in that case, a normal vaccine might not be, or inactivated vaccine might not be very useful. And that's because the normal vaccine is actually more triggered towards producing antibodies, which attacks the pathogens that are staying outside the cell. So when the pathogen is inside the cell, you probably need a cellular immune response as well. And inactivated vaccines are usually not very good in that. in which scenario you need actually attenuated vaccines, which can stimulate both cellular and humoral immunity. But at the same time, like I mentioned before, it can be a very cumbersome process to develop a live attenuated vaccine. Sometimes you might be lucky, but usually it takes a time, and also a safety problem is there. Yep, sorry, I just tried to. Yeah, now I'll go to the next slide. Yeah. So what type of administration? That is another talking point. When you're developing vaccine, you have your vaccine ready, you have the formulation ready as well. You have come to a conclusion, okay, maybe you will use inactivated vaccine or live attenuated, and then you have to think about how do you administer it. There are 3 strategies, at least for the moment. Immersion in a vaccine is a good idea. It's a cost-effective way, in a way, easy to administer. Fish are not that stressed. You just have to crowd them in a tank and then apply it. One key advantage with immersion vaccine is that you can apply it at the earlier stage, when the fish are really small, maybe 5 grams or 2 grams, given that the fish has developed sufficient immune competence. The problem, of course, is it usually appears that it's not long duration of protection when you do this immersion vaccine. You probably need a booster dose. Oral vaccination is not practiced as of today. It's difficult. It's very simple administration, but you are not sure if all the fish are eating the vaccine or getting the vaccine, and then there is a high chance that fish are not protected, because only if some fish are protected, then probably that benefit of the vaccine is not very good. So there are some groups which still working on this, developing this. So maybe it will come soon. Injection is the most tried and tested and effective way of vaccinating fish. Very long-term results. Challenging. It's quite challenging to inject each fish, and you need some labor force. You need better routines to do that. Okay, I move on to another aspect, that is developing a challenge model. Once you have figured out all the other things, initial consideration, you have some of them, what type of vaccine, how are you going to administer the vaccine, etc. And then you focus on developing a challenge model. So what is a challenge model? This is actually the backbone of all your clinical studies when you're developing a vaccine, and that is you have isolated a pathogen from a sick fish, and you want to recreate that disease in your wet lab, and that is a challenge model essentially. So you try to recreate, because then only you can compare your vaccine if it is effective or not, right? So how you challenge the fish is also very interesting. There are different approaches to it. You can have a intraperitoneal injection. You can inject the fish with the challenge isolate in the peritoneal cavity and then wait for it to progress. Maybe the fish dies after 10 days. The same applies to intramuscular injection. You can inject the fish on the dorsal surface and then you wait. You can also immerse the fish, just like vaccination, in the live challenge, in the live isolate solution, and then you will hope that it works. You can also cohabitate the fish. Like what we do there is that you have 30 fish, for instance. You take 5 fish out of it, you inject them in the belly, and then set them with the other fish. So there are 5 fish that are injected that is trying to go within the population, and they will spread the disease. So they will shed after some days and they will die first. And if your model is very accurate or very good, then other fish will start dying or having the disease slowly. So it's very clear that the injection in the belly and creating a disease is kind of artificial. In the field conditions, fish don't get injected, right? So that is not very, I would say it's not mimicking the natural way of infection. And the other end, you have cohabitation, which is very close to a natural infection because the injected fish are spreading the disease to the other naive fish. So ideally, you should have this cohabitation in your challenge model. But that is not easy to develop that model, while injection model is much easier intraperitoneal. So it's a balance of both. You would try to get the other model working because that will give you a better indication of how this vaccine is going to work in the field conditions. So there is an incubation. Initially, when you inject, you have an incubation phase, and so followed by an acute phase and a recovery phase. So this is quite common in the injection in most of the challenge models. Here you see in this graph is on the left-hand side, it's the cumulative mortality. On the x-axis is the days post-challenge. So as you go on with the days, there is some incubation in the beginning, and then the acute phase comes, and then the recovery that I mentioned before. Important things to consider here is that if you get sufficient mortality when you do this challenge, because if you get only 20% mortality, then maybe there's no point in using this model. So there should be sufficient mortality, and you should also know if the clinical signs of this wet lab challenge model is comparable to the field, or if there are some differences. If there are not sufficient mortalities, can you probably have other parameters that you can use? For instance, you can inject the fish with the challenge isolate. You can follow the fish and maybe do a PCR on the fish and see if there is high pathogen load. For instance, that can be a good parameter. If the fish are not dying, at least the amount of pathogen in the body can be a good indication. And there is also a key question of if we can differentiate, if this model is good enough to differentiate between different vaccine doses. So to summarize, a challenge model, you should probably have around 60% mortality, and that could be for statistical purposes if you want to implement mortality as an endpoint in your studies. If there's no sufficient mortality, then you could implement other parameters. You could also use weight information as a surrogate. And always remember that this challenge model is not just a one-time event. You have to keep on fine-tuning it during the process of developing a vaccine. So it's a continuous process. Wherever possible, try to use a model that mimics a natural infection. In this case, it would be cohabitation. Okay, once you have a challenge model ready, you go to the clinical efficacy study. There are some aspects that are useful to have when you are doing an efficacy study, a clinical study with your vaccine. One of that is tagging or marking of the fish. It's just to differentiate between fish groups, not individual fish, I would say. Like vaccinated fish. If you want to put vaccinated fish and non-vaccinated fish in the same tank, then you would ideally want to tag the vaccinated fish, right, separately. How can you do that? You can, in case of salmon, at least you can use fin and maxillae clipping, and they take some time to grow back, so you can have it for a long period, actually. It might not be practicable for other fish for welfare reasons. Maybe the fish doesn't like it. You can use visible implant elastomer as shown in the picture here. It's kind of I think it's a kind of a polymer that can reflect UV light. So you inject it subcutaneously into the fish. For instance, here you can see on the dorsal side of this small fish. On to the right-hand side, you can see on the anal fin of a salmon. And then you have to shine the torch, the UV torch, and you can identify the fish. If you want to identify individual fish, then you can use pit tag. It's kind of a transponder tag that you inject into the belly of the fish. Another important aspect is anesthesia. Fish, when they are getting vaccinated, especially injection vaccinated, you need to anesthetize them. Tricaine is a commonly used anesthetic for this purpose. Sometimes benzocaine is also used. Clove oil is not usually used for vaccination because it doesn't get that deep sedation, but it's very useful in transporting of fish. Important also to have injection devices that serve the purpose. These days in the industry, at least in Norway, they use a lot of type of injectors. As shown in the picture here, for instance, is it has one injection nozzle and two incoming nozzles. So it means that you can combine two vaccines and inject at the same time. You also have a machine injection in some of the advanced farming countries. And usually the vaccine injected per fish is approximately 0.025 to 0.05 ml. Just have a video here of a machine vaccine. Just so the fish are coming on a conveyor belt. The fish are anesthetized and they just are coming in a conveyor belt and they are being injected automatically. So how do you set up an efficacy study? You first inject the vaccine and then and it is divided into, let us say, for example, you have two tanks. Because you use tagging, you can group, test group and control group in the same tank. And this is very useful because then you can essentially remove this tank effect problems that you usually encounter in fish studies. So that's a big advantage. And then you do not have to use a lot of fish for the studies as well. So you can have two groups here. One is vaccinated with your test vaccine. Another is vaccinated with maybe a controlled substance like a PBS or even the adjuvant that you are using for this vaccine. So let's say, for instance, here we have 60 fish for each group and for each tank. You inject them accordingly. Your test group gets a vaccine, the control group gets the PBS, and then you let them go back to the tank. What you do is you take maybe 10 fish, anesthetize them, do the marking, give the injection, put it back. So put it back or maybe put it in another tank so that you have control over it. That way you vaccinate all of these groups and then you have an immunization period. And this varies and that you have to figure out based on your experiments, how long you have to use it. In the case of salmon, it's usually 6 weeks of immunization period. That is by standard used. In case of tilapia, 3 to 4 weeks is quite enough. And then after 6 weeks, you have your challenge model. So you follow that. You split this fish into 4 tanks, maybe. And then you can use 2 challenge dose. One set of tank gets a challenge dose 1, and another set of tank gets challenge dose 2. So you try to inject in 2 challenge doses because there could be sometimes you don't get high mortality, high death rate, and then you probably have another dose just to cover it. So what comes out of this is that you are injecting the fish with a pathogen after they are vaccinated and after for the immunization period is over. And then you get you can plot the death mortality in the graph. And for example, here you can see the control fish can die faster and they are almost 80% mortality. At the same time, the vaccinated fish are maybe having 20% mortality. So this is the difference. This is what your efficacy is, and that is measured as a relative person survival, RPS. That is kind of a term that is used to denote this efficacy of the vaccine against the control. What you can take out of this study is that you can also get some antibody related data. For example, after vaccination and after 6 weeks or after the immunization period, you can take the blood samples and try to plot and do an ELISA to know how much antibody is present after your vaccination. And you can maybe compare. You can see that vaccinated fish have higher antibody titer. So that's a good measure of efficacy, but this is also, this again needs fine tuning. It has to be consistent, etc. So it would need a lot of work to get this assay. You can also take histopathology and do necropsy of the fish. It's very interesting to know that the fish are not dying in this vaccine group, but what about the other fish? Are they affected in some way? That's also a good question, right? So then you have to follow through with histology to see if the surviving fish are okay, or they were just like hanging in there, like, okay, almost going to die, but not yet dying, right? So it's very good to know all of these aspects in when you're doing an efficacy study. I just mentioned RPS and how it is calculated. It's calculated with when you compare with the control group. It's a measure of efficacy of a particular vaccine or an antigen in some cases. Usually an RPS of greater than 60% is desirable. And here I have an example. For instance, this is taken from a product label of one of our vaccine. You can see that it's a multi-component vaccine. That one shot gives a lot of antigens, like 6 antigens. And for each antigen, we have done a test, or Pharmac has done a test to identify what is the RPS. So it varies for each antigen. Some have a low, some have very high, etc. So this has to be provided as an information. And last but not least, efficacy study is not just this. There are a lot of type of efficacy studies that you have to do in the process of developing a vaccine. And that includes duration of immunity, how long it lasts. So you can increase the immunization period to 20 weeks, for instance, and challenge after 20 weeks and see how the protection is. You have to do a dose finding study. What dose to use is a big question of vaccine. Usually you start with a ballpark figure based on your experience, and then you have to fine tune it. So it's important that the challenge model is able to distinguish the same. You also have to figure out onset of immunity. That, for instance, if you are, for example, doing a bath vaccine, it would be good to know if, or not even bath vaccine, it applies to all vaccine. It would be good to know how fast the fish is getting immunized. For example, is it already immunized after 1 week? Then it's great. Then the farmer can already put them into the pond, or for instance, or expose them to the normal world. So that helps. Yeah, and it's followed usually by safety studies as well. I mentioned inactivated vaccines as very problem in the sense it's very good, effective, but it also has another side to it, and that's because you have an oil component and that's deposited in the belly, etc. You get a lot of side effects. Sometimes there will be local reactions like this, adhesions. But modern vaccines, these things are reduced considerably. So how that is addressed is by using a much lower volume of injection. So these things have to be figured out. In the case of live attenuated vaccine, of course, you have to do a safety to the fish test. And if it's reverting to virulence after you inject, after several cycles, if it is reverting to virulence, etc. Local reaction problems are usually much less in this case. And then we go on, after you have completed all your safety and efficacy studies, it's time to test it in the field before you go for the license. And this study is very important because then you are approaching a farmer and saying, okay, I need my vaccine tested. Can you cooperate? In that case, the farmer will agree to do the test. Fish will be his. He will just take your vaccine and test it on some fish. And then you need some good statistics and good study setup. And usually internationally, you follow a good clinical practice principle to do this field test. Very important that the site you select, the farmer you select, he is suffering from that problem. It's no use if he, or at least minimal use if that place doesn't have the disease. And also this farm should have access to good quality seeds. Farm personnel should be cooperative and they should know how to record data, etc., and keep the track of the data. That's also very important. That's actually part of the good clinical practice. And not the least, farm should have good routines and biosecurity measures. And once you have done all of this, your product is licensed, hopefully, and it's available in the market, and still you are supposed to follow it up using pharmacovigilance. That means that you are you're continuously gathering information on the use of your vaccine. Is the effect as promised? You should try to get feedback from the farmers. How is the side effect profile? Is there any issue with efficacy, underperformance, other aspects? How long is it effective? That's also good information that you can add later into your license if you get good feedback. And it's also important to keep track of if the disease variants are coming. If you are just like COVID, if there is a new type of variant coming, will that be effective against your vaccine, or should you go back and change your vaccine? Just giving some examples here. These are, for example, currently a lot of vaccines are available for salmon. You have here a 6-component vaccine for salmon. Below that, there is a 1-component vaccine against pancreatic disease. You have a live attenuated salmonid rickettsial septicemia vaccine, which is very effective and equally effective as an inactivated vaccine. That's been a very good product. And recently, Europe, at least in Europe, they have approved the first DNA vaccine for fish, and that is a salmonid alphavirus vaccine made by Elanco. Yeah, so there are a lot of vaccines. If you get in other fish species, there are much less. In European sea bass, you have a VNN vaccine, which is quite effective. In striped catfish in Vietnam, you have reasonably performing a very good vaccine, actually, Punga 2 against Ictaluri and Aeromonas. In tilapia, streptococcus is a big problem, so there is a vaccine against it as well. Yeah, so I just thought of giving some 1 or 2 short videos, very short, from that's kind of a promotional video from Pharmac, but at least it gives you some idea about the vaccination process. In this case, it is CBUS. Just one more short video, and then we are finished. Yes, thank you. Sajana, the floor is yours.
Okay. Thank you, Binoy, for delivering an excellent talk. And thanks to all who joined today for the talk. Before we go to discussion or leave the party, please provide us with the feedback about today's talk. The link to the feedback is given in the chat box. We are always looking to expand our coordinator network. So if you can help us circulate our talks, we would like to hear from you. Just send us an email to afstalks@gmail.com. Now let us move into the discussion part. We have 15 minutes for discussion. Please use the hand raise feature if you have any question to the speaker, or you will be given a chance to speak. If possible, please also type the question in the chat box so everyone, including the speaker, can read and understand your question. Let me check whether we have any questions in the chat box. Yeah, there are some questions. Yeah, there's a question from someone whose profile name is My Dream. He has asked, please, where can I get tagging gun if someone know website? If it's available here, he can directly ask also. I think he is meaning tagging gun. Tagging gun.
Tagging gun.
Gun, maybe gun, that equipment to.
Yeah, I think I assume it's in India, so I'm not sure if somebody is providing it there. So it depends if you have a local supplier. It's usually, for example, a German company called Henke Salzwolf that is providing. I can write it down. It's a German company. But I'm not sure if it's available everywhere.
Okay. I have some query. Means it is very simple thing. We do vaccine efficacy trials, as you mentioned. No? So Norway is a cold temperate country. So apart from salmons, you do for sea bass and also this pangasius, which are cultured in tropics also. So if whether you do the trials in different climatic zones or whatever you do.
I know some people trying to do tilapia thing in Norway, but then they have to keep the temperature high, and it's a lot of energy required to warm the water, etc. But we usually do in different parts of the world, so we have contract labs that we can do.
Okay.
At least, so that's how usually we do.
So the vaccines are standardized for tropics also, like it is trial has been done in the.
Yeah, of course you have to do the trial in the fish, on that particular fish, and I think that's how all the companies work, that if they want to make vaccine for a warm water fish, they have to either go in the warm water part warm parts of Europe, for example, Spain, Greece, they also have sea bass there, European sea bass.
Okay.
Or we also have office in Vietnam, so we go there and do the trials.
Yes.
Yeah.
I think Deepak have a question. He has typed in the chat box. Deepak, will you ask it directly?
Hi, Binoy.
Hi.
Yeah, I have written it pretty detailed in the chat box. Yeah, wonderful presentation. Because it's different from a research or academic talk. You have given the insight from a real world perspective. How what are the problems when you actually develop a vaccine? So very good at that thing. So my question is about. I'm just wondering, what about shellfish or oysters? Is there any leads into that? Is there any vaccine development? Regardless whether there's a vaccine or not, what is your take in the problems or hurdles in developing a vaccine for them, and problems in administrating them? Because many times, whatever disease it is, the response is by, for oysters, it will be like gaping shells, and it will be too late when they are unhealthy. Unlike the fish, which you can see more symptoms when they are alive. So for oysters, it will be like close to death, and it will be too late to respond for anything. So what do you think about the vaccine development for oysters?
Yeah, I think it's comparable to shrimp, isn't it? Like in a way where they are placed in the animal kingdom. So the question is whether oysters can have an adaptive immune response to vaccination. And at least a lot of studies have been done in shrimp, which doesn't, or at least we believe because it's an invertebrate, it doesn't have an adaptive response, so it cannot be theoretically vaccinated. But in shrimp, the studies have been so advanced. There is a lot of studies in shrimp, and at least some scientists have been able to show that there is some kind of a response. Like it's not like an antibody response or anything, but there is some localized response that can help for maybe 1 month or so, but then it vanishes. So it's difficult to answer, Deepak. I think it's a good area where prophylaxis is important. Like you mentioned, it's very required, but from a vaccine perspective, it seems, or at least from my thought process, it's one idea could be having a bath with a vaccine, but we don't know how long that effect could last. Maybe you would be able to stimulate innate immunity, but that can last for 2 weeks, but not necessarily a long duration protection.
Thanks, Binoy. Yeah. No worries.
Somebody was asking, what about the economy of fish vaccine? Whether it's feasible for farmers to afford in country like India in future? That's, I think, the very good question. I am, as you know, I'm working in Norway, so I'm not really very familiar, or at least I don't know the current details, what's happening there in India with respect to farming. It's a very good question, especially if you're thinking about normal traditional species of fish in India, like carp, or other, low value, so-called low value fish, but it's unthinkable maybe to inject them, because then you are spending a lot of money. When salmon, when you think about salmon industry, what does a vaccine cost? You set, for example, vaccine should be around 1% to 5% of the per kilo cost of the fish. So if you have a 40 kilogram, if fish that fetches 40 kilogram per 40 rupees per kilogram, then your vaccine dose per fish could be around 40 paise, 50 paise, like that. But it's the thing is that what kind of fish we can apply that. Maybe sea bass is an upcoming. I know it's a lot of future talk about sea bass in India. If that really catches on in the cages, etc., then I think vaccination would be very, very important. And equally important, like I mentioned in that my talk, is the access to good quality seed, and that is very, very decisive, I think. I'm not an economic expert, but at least this is what I feel. Please provide complete list of vaccines. Sajana, it's okay if I read this through, or do you want to? Sorry.
Yeah, Binoy, you can read out it.
Okay. I don't know. Please provide a complete list of vaccines.
Binoy, someone asked contact email for you. Can you provide your email address in the chat box?
Yeah. Okay, maybe I'm answering to just some person. Yeah. Somebody was asking, I would like to know your opinion on whether fish mucus mediated nanoparticles can be potent antibiotics replacement. I'm not sure if I got that question.
Who asked the question?
It was Phoebe.
Yeah, Phoebe, you can unmute and you can talk.
Yeah, hello, Dr. Binoy Rajan. So I was asking, so when Sajana ma'am introduced you, she was saying that you had some prior expertise in fish mucus, you worked on fish mucus. So I was just wondering that whether we can extract fish mucus from low value species like common carp, and that is available in India, and we can, there has been studies like fish mucus has some antibacterial property. So we can increase the efficiency and we can connect them with some nanoparticles so that we can deliver them as a delivery method. So I'm thinking whether that can be doing some, giving some general immunity.
You mean to say administer them as like feed or?
Yeah, it can be in any way. It can be like.
Yeah, okay. No, I'm sorry, but I haven't thought about that angle because, yeah, one thing is to get enough mucus. I don't know how that would be. Either that, or maybe even a better way could be I isolate or identify some components in the mucus and then try to synthesize them to scale up, or could be a good try. But yeah, just like I think the antibiotic era, it's like just a trial and error. So I think it's an explorative idea. It sounds good. But how can you collect a lot of mucus, and is this a viable option, etc.? Maybe a good idea to start with potency testing in the lab, like in the cell culture first, before you do other things, so that you know what's happening there.
Jomol also has some question. Jomol will ask us directly. Vaccination possible only through injection, no chance to give it through feed. I think it already explained.
Injection and immersion is a good idea. It works. Injection used to work much better because you are essentially putting it inside the fish. Immersion works. Usually experience is that it doesn't last long enough. Then you have to give a booster. And the feed aspect I mentioned, it's nobody has managed to figure out that yet. And I think there are difficulties in administration, like all the fish probably will not get the same dose if you are giving through feed. And that's a disadvantage. Yeah.
And one another question from her is that is vaccination available for tilapia lake virus?
Not yet.
Okay. Okay. Pradesh has already asked me. There is another question from BG to everyone. BG. In future coming years, among both finfish and shellfish, which has more scope in case of culture or market, etc., throughout worldwide? But if he is available, he can directly ask the question to Bina.
Yeah, I think it's a very general question. It's shellfish. It's difficult for me to answer. Finfish, I think it's very difficult to say. Tilapia has been touted as the big takeover, like it's going to spread everywhere. But it also depends if let's say in India, for example, we have a lot of small scale farmers, for example, a biofloc bloom. So there have been a lot of people practicing this biofloc methodology, but they're all very small scale. So it would probably start with some big company coming in and consolidating a lot of this and starting a big business, maybe focused on export, etc. So then it would determine the fate of that species. Tilapia seems to be the best positioned fish to take over, at least in terms of volume, but I'm not sure about other fish types. Yeah.
Okay, I don't see much more questions here. If anybody has questions, please, they can directly come and ask. The chat box, I don't find more questions. Thanks, Binoy. It was a very interesting
Thank
session.
you.
I hope there is no more questions. Let me check the inbox once more.
Can I also add a disclaimer, so to say, just because I work for a company now, so I am not presenting this on behalf of that company. It's just from my personal capacity. So that that's yeah.
Okay.
Yeah.
There was a question from Pradesh.
Did
you?
Hey, Binoy, I think you almost answered that question. That's why I didn't.
The question from yeah, the similar question.
Yeah,
Okay.
it is a cost question, but again, like seems like this is more appropriate for like a big industry than a small or mid-sized farmers, right? Is it
Yeah.
something farmers of a small capacity can utilize this one? And that's my question.
There is also, like I mentioned, a lot of long process involved in this development of a vaccine, but there are alternatives as well. Some regulatory authorities allow to develop a vaccine if they face some problems in some particular area. For example, in a small area, for example, in India, if in Mumbai or in Maharashtra area, you have a lot of disease problem with some particular disease, then the authorities can say, okay, you don't need to develop a lot of things, just make a small vaccine that can be used only in that area. In that case, the developmental cost can be very little or maybe much less, and then you can sell for a lesser price. I think some authorities do that. Maybe Pradesh, I think might be familiar with that as well. It's I think it's autogenous vaccines, they are called. That's like a stopgap solution. It's not like you can take it to another place and sell it. You have to sell it there, actually.
Okay.
A few more questions are there in the chat box now. One from Saurav. He has asked, which one will be the best and cost-effective option, vaccination or disease treatment after infection?
Yeah, I think that is we are living through that phase now, isn't it? What would you want to be treated for COVID, or would you vaccinate against it? I think the treatment aspect, the problem is that treatment has a lot of uncertainties. That is the problem, actually. Vaccinated, yes, you can still you might get a disease outbreak after vaccination, but still you have to see it as an insurance policy. So it's difficult to say. If the disease, you cannot develop a vaccine, then it's justified that you probably look for treatment. But yeah, I would suggest vaccination all the time.
There is a question from Divyanand. Whether he wants to ask it directly, he can ask. For humans, we have clinical tests for vaccine. What about fishes? They do have any clinical trials? Whether the vaccine approved by any organization?
That's exactly what I have gone through today. You have to do clinical trials, then you have to submit all the data to an authority. In India, maybe at least there will be some authority. I think it could be a animal husbandry department or something.
He's asking whether global level we have any other such organization.
No, at least in Europe, it's the European Union that covers a lot of countries. They have an authority, for instance.
Okay.
But I am not familiar with a global, because even if it's globally approved, it doesn't necessarily get approved in each country, so it doesn't matter.
And a question from Vishwajit Mishra. He has asked how a farmer come to know that fish need vaccination.
Yeah, it has to be a recurring problem. If a farmer faces a problem every time, and if he thinks that the disease is not going away, and also it depends on the dialogue between a farmer and an extension officer or an academic, what information he can give the farmer. So I think it depends a little bit on those kind of interactions. It's usually a combination of both farmers and academics who take this initiative. It's not either of them just alone. A farmer comes with a problem and then academic works on it, right? That's how it works. Yeah.
I don't see any more questions here in the chat box. Yes, one more question asked from Debashish Jaina. Is natural immunity better than vaccine acquired immunity?
Better in what way? In the sense,
if Debashish is available, he can clear the question. Otherwise, he can answer.
Not here.
No.
It's an interesting question, but I think we have to see what we lose when we get a natural immunity. Do we lose something? Do we lose our, in case of humans, if you lose a lot of your, or if there is an organ failure and then you survive and then you get a natural immunity, then it's no point, right? So it same applies to fish as well. I don't know how you have imagined natural immunity in fish, that just let them get infected, or if it's like that, then of course you will lose a lot of fish in that process. So yeah, so it depends. I don't think natural immunity is it's a strategy used by anyone. No.
Phoebe has come up with one more question. Is Phoebe asking it directly? Are you asking it? Okay, I was helping.
Yeah. So I was just wondering about this exact. I'm not aware of the exact mechanism how the mechanism works. So generally, just like when Deepak sir was mentioning about like our lower invertebrates, or like our shellfish doesn't have a like memory cells, or they don't have a specific immunity. So if we have some exploration or insights on how exactly they work, so we can try to implement or create this in some lower invertebrates, so that they can also have all the ability to have the specific immunity. So what are the latest studies? That's what I'm asking. The extracellular mechanisms.
I don't really know about what are the recent studies, but it's a lineage of cells, the memory cells that is found in vertebrates, in human and in lower vertebrates, that is not present in this low invertebrates. So that's the reason. And there's also some important genes like the RAG genes or something like that that's not present, I think. But what can we do to go around it? It's I think many scientists are trying to figure that out. And especially, as I mentioned, shrimp is a good point to get all of this information, because shrimp studies, a lot of advancements are made there. People have worked with several type of things, RNA, what is that? RNA interference strategies, etc. That could be a good idea, actually, instead of vaccination. So it's not that you have to think in the same way. You can go around with some other strategies that could be useful. But unfortunately, I don't really have a lot of information in my head right now. But I think a lot of information is available with respect to shrimp studies, especially comparing shrimp with fish, etc., shrimp with humans. That probably you could get some info there. Yeah.
Okay. I don't find any more questions. I guess that is the last question. Thank you, Binoy, for the interesting discussion section and interesting talk.
Thank you. It's been a pleasure. It's very exciting to have this discussion and etc. So I think I would like to attend a future session with other speakers as well. It's quite nice.
Okay, you're welcome. And thank you for the wonderful session. Thank you very much.
Thank you. Thank you.